Surface plasmon resonance is a very common technique used by biotech companies and pharmaceuticals to determine the kinetic parameters of a product that they've created. And this is important because if you are trying to sell a product, you need to go through the FDA clinical trials, you've got to have data to quantify how much of a binding affinity your particular compound will have in vivo, for instance. And so the KD, the dissociation constant, will tell us how readily our ligand, our product, will dissociate from some protein that we've designed our ligand around. And so what we get out of an SPR machine is the raw data that we get out is this plot that tells us how R, a resonance unit, is evolving with respect to time. And so what's happening in the SPR machine is we are gradually increasing, or we are flowing a constant concentration of ligand or protein through a matrix that has our protein in it. And what happens is the binding sites are becoming occupied, and once all of the binding sites become saturated, when they're all full, you no longer are seeing a change in your resonance units over time. And at that point, we know it's saturated. We will begin to allow, where we will flow solvent through our system and allow the ligand and the protein to dissociate from each other. And so it is this region here that we are interested in determining what's going on here. So we want to find what KD is, and we will do that via the following. And so the first thing we're going to turn to is examining this equation here. We've got a protein and a ligand binding together or unbinding to form a protein ligand complex. And so what we can say is that the rate of change of the concentration, which is these square brackets of the protein ligand complex with respect to time, is equivalent to minus KD times the concentration of the protein ligand complex. And we need this minus sign out front because KD must be a positive value. It's a kinetic parameter. It has units of per time, perhaps. And PL is also a positive value because it has units of concentration. And so what we're going to note is that the concentration of bound ligand, PL, is proportional to the resonance units, R, in this y-axis of our plot. And so what we can do is we can make this substitution and say that it is dr dt that is equal to minus KD times PL. And if we also make the other substitution and say that KPL on the other side was also equal to R, we can rearrange this equation to get it into the form dr over R is equal to minus KD dt. I'm sorry for going off the page. And this is using the method referred to as separation of variables in calculus. And so if we now integrate both sides of our equation, we will integrate from R naught to R, from T naught to T. We'll recall that the definition of the integral of 1 over x is equal to the natural log of x. So what this equation turns into is natural log of R over R naught is equal to minus KD times the anytime t minus the initial time t naught. And so t naught would be right here on this plot, and R naught is R evaluated at t naught is equal to R naught. And so with this information, we will know we can take several points along this line. we would know T at that point. We would know that R at that point. R naught and T naught are constants, and we could solve for KD. And we could also linearize this equation as well if we wanted to take a slope and average all of the points of data that we have to obtain a more precise final answer. And so with this value and moving around a negative sign, we will have found what our dissociation constant is for our particular protein and our ligand. And so this is very important stuff if you are interested in biotechnology or how a molecule will, or a ligand will interact with a protein, how readily it will let go. Oftentimes when we're evaluating candidates for drugs, you want stuff that will have a high binding affinity so that it can have a long half-life inside the body, for instance, so that it can be effective and potent. But it is pretty interesting stuff to think about. I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.